So we are presenting on the water and salt balance systems of the termite.
Osmoregulation is the active process of maintaining salt and water balance across membranes within body fluids. It is a process necessary to keep the body fluids from being too diluted or too concentrated. Termites are terrestrial insects, and most terrestrial insects are osmoregulators, which means that they must maintain the right concentration of solutes and the right amount of water in their body fluids.
So insects generally regulate their water balance through selective reabsorption. They have Malpighian tubules, which form a primary excretory fluid made by the active secretion of ions and the water that passively diffuses with it. Osmotic regulation can then be achieved through selective reabsorption. Termites can also control water balance by drinking water from the wet soil in their mounds, as well as storing water in their crops. Termites can also utilize their mounds for water regulation. In the rainy season, they build specific water channels that lead the water to the outer walls of the mound. But then in the dry season, the termite mound reduces evaporation, and sometimes the termites can even actively transport water to keep the insides of the mounds moist.
Malpighian tubules generate urine flow that is isoosmotic to hemolymph that contains potassium chloride and sodium chloride. Active cation transport occurs across the Malpighian tubule principal cells, which are essentially Malpighian epithelial cells. These cells contain long, thin apical microvilli, each housing a mitochondrion. Cation transport at the apical membrane is driven via a baphyllomycin A1 sensitive V-type proton ATPase pump, which is primarily expressed at the apical brush border of the principal cells. This pump allows for the return of protons to the cell via a proton gradient, which takes place by means of amiloride-sensitive cation and proton antiporters. At the basal membrane, potassium uptake takes place via bumetanide-sensitive NKCC cotransport, which occurs due to the favorable sodium gradient for entry into the cell. This sodium gradient is managed by basal membrane sodium-potassium ATPase. There's a favorable transepithelial electrochemical gradient for chloride to enter the lumen via passive diffusion by means of either paracellular or transcellular conductance pathways. Diuretic hormones stimulate net KCl and NaCl movement into Malpighian tubule lumens and have no large effect on osmotic permeability. This remains even true when fluid secretion is elevated by 1,000-fold.
So termites are not likely to move from an environment that isn't similar to their current one. They are ectotherms, therefore external factors will impact their internal temperatures. If the weather surpasses the abilities of the mound to keep them warm, then the termites will die. According to a study on the low temperature effects on survival of the eastern subterranean termite, it was found that worker termites exposed to 0 degrees Celsius for 10 days exhibited high mortality rates regardless of a duration of 10 degrees Celsius acclimation periods.
So termites do not have kidneys. Instead, like most insects, they have Malpighian tubes. These are structures that precede the rectum in the termite's digestive system, and all of the termites' undigested food passes through the Malpighian tubes, unlike the human kidney. In the Malpighian tubes, there isn't any hydrostatic pressure since there isn't any blood, so kidney-like filtration cannot occur. Instead, the walls of the Malpighian tubes allow for osmosis and diffusion directly with the hemolymph. Ions are actively transported into the Malpighian tubes, and water follows those ions. This results in the production of a urine-like substance. Much of the water is then reabsorbed in the rectum, but the products that aren't ultimately reabsorbed are released from the termite as waste. Like human kidneys, the Malpighian tubes are controlled hormonally, but these processes are not particularly well understood.